friends are trying to join. Maybe because of network issue, they are facing problem. If that is so, then we'll start with our class. I hope you all are here, not losing your tolerance. Be patient for a while, okay? Okay, so shall we begin? Yes, those who are in the Google Meet, can we start with our class? Yes?
Yes,
Yes?
sir.
Okay,
Yes,
it's
sir.
better to start. Otherwise, waiting for others will be taking time, and who knows? I'll be also recording the same lesson so that they can watch the video also. So, while teaching, I'm also recording the same in the YouTube. Okay. So in the morning, we stopped with this slide saying that we are having three basic steps. Though we are having so many steps, we can say that we can have three basic steps, or the three main or three key steps, so that through those three key steps we can genetically engineer an organism. We can genetically modify an organism. Particularly, we are applying the concept of genetic engineering in production of different food crops or in production of different plants before practicing it into the animals. Okay, we are using genetically modified organism, particularly genetically modified plants, in order to produce different agricultural crops. So in the morning, I was just briefly telling you about how that process is done. So here we have a picture showing us about the crop genetic engineering process. First, what I said is that suppose if we are having a potato, and in that potato it is having lots of starch. So this potato is having lots of starch, and that characteristic, the character of having lots of starch, is due to a gene, right? So that gene, if we could isolate, then that gene is going to be a desirable gene, right? So our gene of interest, which was extracted from the potato, which is rich in starch, is extracted. That is going to be a desirable gene. Okay, so the first step is identification of gene of interest or desirable gene, the gene that we are required from the particular source of gene. Okay, after doing the isolation of gene of interest, we should try to introduce the gene of interest which was identified into the host organism. Now here the host organism or host plant here is maize or corn is, you suppose if we are not having a corn which is rich in starch, so we can introduce the gene of starch producing gene which is obtained from potato into the cell of maize plant. Okay, and that process of introducing the gene of interest which is obtained from the potato, and this gene of interest will be producing lots of starch. And this gene of interest, after introduction or after transformation or after injecting into the cell of maize plant, then that process is usually what stated here: introduction of identified DNA into the host. Okay, the host here is cell of maize plant. And after introduction, it's important for the genetic engineers to maintain the same gene within the first generation of plants. Even after reproduction of the same plant, again that gene should be maintained. So the third step is maintenance or keeping the same introduced gene in the host plant even after 4 5 generations. Okay, and if we can do that, then that is achieving how we can genetically engineer an organism. Okay, particularly here we are trying to look at the genetic engineering of maize plant. Okay, so same is the case if we are having a gene which is resistant to insect. Suppose we are having a gene which is isolated from bacteria and it is showing resistance character towards the insect, then if we could introduce this gene which is showing resistance towards the insect into the apple fruit or apple plant, then that apple plant will be genetically modified, right? We are introducing a new gene, and that gene is from bacteria, and that gene will be giving apple tree resistance to insects. And that kind of process is modification of an organism. Particularly here we are having apple tree, right? Apple plant, right? And in a way that apple will be resistant to insects. Okay, so hope you all are following what I'm trying to say here. I hope 3 points are clear to you all.
Yes, sir.
Yes. So there are three main points. First is identification and looking for the gene which is uh, desired, the gene of interest. After isolating it, then we'll be introducing into the host cell. After introduction into the host cell, those host cell after multiplication, after cell division, and even after the next generation, if we could maintain them for say like 5 to 6 generations, then we are achieving the technique of genetically modifying an organism. Okay, so this is a simple three-step based introduction about how we are genetically engineering an organism. Now, in the morning and yesterday, we have seen the animation of how we are uh, following up with all the processes and principles of genetic engineering. And we said that we are trying to introduce a piece of gene. Even here also we are trying to introduce a piece of gene, right? Let me look at the cursor and let me first find the laser. See, we are having a gene of interest which is introduced into the host cell. Same is the case here also. We are trying to find the gene from bacteria. After identification of the desirable gene from the bacteria, that gene which is resistant to insect is introduced into the cell of the apple tree, right? And in a way we are able to produce newly engineered, genetically engineered or plants, right? So what is going to happen with a piece of gene? We are seeing that there is introduction of piece of gene or piece of DNA, and uh, now here we are looking at a piece of gene, and that piece of gene is going to follow up with the certain procedures. So the question here is, what is the fate of the piece of foreign DNA which is transferred into the alien organism? And it's written here, the piece of DNA would not be able to multiply itself. Okay, this is the condition. uh, in the organism there is no multiplication of that piece of gene alone. So if that is the condition, that DNA is not going to multiply, that DNA is not going to integrate or join with the host cell DNA. So what is the fate of the piece of foreign DNA which is transferred into alien organism? So that is what we have to understand. So first we said that we have a piece of gene, right? Piece of DNA, and that is called as gene of interest. Sometimes we also use term foreign DNA, and even we also use alien DNA. So we are having alien DNA or foreign DNA or gene of interest or desirable gene, and that gene we have to introduce into the host organism. Now we said that there is no that gene is not going to divide, that gene is not going to have impact in the host cell without depending on other gene. So we are saying that piece of DNA get integrated into the genome of the recipient, okay, particularly at the ori site. Ori here refers to origin of replication. So in the host cell genome, in the nucleus of the cell, host cell, we are having DNA, and that is usually called as genome. So DNA is having a region or a segment called ori site. Ori here refers to origin of replication. We are going to discuss about ori site in the coming upcoming topics also. Okay, so here what we have to understand here is a piece of gene that is called as desirable gene or gene of interest or foreign DNA or alien DNA should be joining with the genome of the host organism, particularly at the oocyte. And in a way, there is multiplication or cloning of the gene of interest occurs. So, cloning here is making multiple copies of the same DNA, the same desired DNA occurs. So, this is what we can say. And that piece of DNA can be inherited along with the host cell DNA. So, we can say that the fate or the luck or what is going to happen with a piece of foreign DNA or piece of desirable DNA which is introduced into the host cell organism is, first there is integration or joining of the gene of interest with the host cell genome at oocyte. And after joining at oocyte, you have studied earlier in the earlier chapters, you may have studied and you may be remembering. Oocyte is the region where there is formation of replication form, right? And at oocyte, then there is cloning occurs, cloning or multiplication, a replication of the DNA occurs. And in a way, we are going to get a new gene which is having host cell DNA along with the gene of interest and which can be inherited in the next generation also. Though the concept here is not so easy to understand, I hope you are getting some idea regarding how after introduction of desirable gene into the host cell DNA or host cell nucleus, what is going to happen with the gene of interest. Here we are saying that after introduction of gene of interest into the host cell, in the host cell, there is also DNA of the host cell, right? And to the DNA of the host cell or genome of the host cell, there is joining at the oocyte. After that, there is multiplication or cloning of the gene of interest along with the host cell DNA. And in a way, that piece of DNA which is desirable for us will be inherited along with the host DNA as the host cell multiplies or host cell undergoes cell division. I hope you all are getting a brief idea about what is happening with the piece of foreign DNA which is transferred into the alien organism or alien cell. Are you all getting some idea? Yes?
Yes, yes, sir.
So even if you are not able to understand here, kindly just follow this flow chart. Okay, and in a way, by studying, then you may be able to understand. And after continuous reading and continuous explanation by me or even by some other will help you to understand, because these techniques are not practiced by us. We can't practice here, right? And in a way, these are higher level uh, experimental procedures which we can't even explain here. So just trying to follow up the information. Okay, so kindly just go through the flow chart properly later, and then you may be able to understand. Okay, now let's look at the tools of our DNA technology. Earlier we already seen the animation and we was knowing different tools which are being used in our DNA technology or genetic engineering, right? So in the recombinant DNA technology or rDNA technology, we are using some instrument or equipments or tools, particularly enzymes and all, right? So first tool that we are going to use in genetic engineering is enzymes. We have different types of enzymes, right? We have seen ligase, we have seen endonuclease, right? So we have nuclease or restriction endonuclease enzyme, which will be cutting the DNA at the restriction site or recognition site. Okay, so we have enzymes called nucleases enzyme, which will be cutting the DNA. Okay, for them we are also calling them as restriction enzymes, and restriction enzymes cut DNA at recognition site or restriction site. We also have heard about ligase, which is helping in process of ligation. Ligase will be joining two DNA, two cut sticky ends of DNA. Okay, we are also having polymerase enzymes which are used in our DNA technology, particularly in the PCR, polymerase chain reaction. Okay, we'll be able to understand more once we go for further discussion. Okay, vectors, we have already seen that we had already seen the plasmid vector, right? Like plasmid vector, there are also other DNA which helps in carrying the gene of interest. So earlier vector, the term vector is mainly referring to the carrier, right? We have seen plasmid vector. Plasmid here is small circular DNA, right? And plasmid vector will be carrying the gene of interest, and in a way we can say plasmid DNA is a vector. Similarly, we are also having other vectors which are obtained from bacteriophages. Okay, and we also bacteriophages can be also acting as carrier of the gene. We also have cosmids which helps in transfer of piece of DNA. So vectors are mainly responsible for transfer or carrying of piece of DNA which is attached to it, right? We have seen the animation earlier, so hope you all are able to follow up what we are trying to understand here. We also have seen the E. coli as host cell or host organism into which we have introduced our DNA, right? So these are some of the tools which are involved in our DNA technology. These are one of the key tools or requirements for doing genetic engineering work. Okay, in addition to these different tools, we also require sophisticated instruments. Okay, are you all able to understand? Yes?
Yes, sir.
Okay. So we'll proceed further. Now, earlier we said that we require restriction enzymes, right? And enzymes are of different types, right? And particularly the restriction enzyme that we have used in our DNA technology are also known as molecular Caesar. Why molecular Caesar? DNA is a molecule, right? Biomolecule, right? RNA is also a biomolecules. So restriction enzymes helps in cutting the DNA. That's why they are also called as molecular Caesar. Like Caesar, which helps in cutting different material, right? Cloth, paper, right? Same is the case. Restriction enzyme helps in cutting a molecule, particularly your DNA in genetic engineering. So that's why they are also called as molecular Caesar. Usually, restriction enzymes are present in the bacteria, and these restriction enzymes will be defending the bacteria from bacteriophage. Bacteriophage is a virus which usually attack or eats the bacteria, and hence, in order to protect bacteria themselves, they produce enzyme called restriction enzyme, and that restriction enzymes will be killing the bacteriophage, killing or preventing the life cycle of bacteriophage. How bacteria are having restriction enzyme, and those restriction enzymes will be cutting the DNA of bacteriophage, which is injected into the bacteria. And by cutting the DNA of bacteriophage into bacteriophage, then there is neutralizing of the DNA of bacteriophage, and hence life cycle of bacteriophage couldn't occur. I hope you do remember the life cycle of bacteriophages. Bacteriophages are viruses which are attacking bacteria by injecting their DNA, right? And suppose if bacteria is having restriction enzyme, and if the bacteriophage had already injected the DNA into the bacteria, then these bacteria can release restriction enzyme, and restriction enzyme will be cutting or digesting the DNA injected by bacteriophages. Hence, bacteria themselves are protecting or defending against infection or attack of bacteriophages. Okay, so usually these restriction enzymes, now with reference to such mechanism, we can use restriction enzyme in our DNA technology, genetic engineering processes, right? So we said that restriction enzymes are responsible for cutting DNA, and restriction enzyme will be cutting the DNA at particular site or particular region of DNA called recognition sequence. Recognition sequence are usually the sequence of base pairs which are recognized by restriction enzymes. Our DNA is having sequence or order, or there are many base pairs, nitrogenous base pairs, one after another, right? A, T, G, C. So these sequence of base pairs uh, are recognized, first observed, identified, and cut by the restriction enzyme at recognition site or recognition sequence. These are even also called as uh, restriction site, okay, as restriction enzymes are cutting. So after cutting the DNA at recognition site or restriction site, then we are going to get sticky ends of DNA. Sticky ends means ends of the DNA which can join easily. Sticky means something which can stick easily, something which can join easily, right? And see, the collective term used for restriction enzyme is nucleases. So we have different types of nucleases. If we look at the types of the nucleases, we have exonucleases. Exo means outside, right? And nucleases means enzymes which are digesting DNA or RNA. So we are mainly discussing about genetic engineering, where we are trying to engineer the DNA, right? So restriction enzyme which is cutting the DNA at the ends of the DNA. See, we are having one strand here, DNA, a DNA which is having 5 prime to 3 prime and 3 prime to 5 prime strand. These two strands are forming a DNA. If we add exonucleases, then these exonucleases will be cutting the DNA at the ends. See, there is cutting of the DNA at 5 prime to 3 prime ends. There is no cutting in between. But in case of endonucleases, endo means inside. So within, suppose if we introduce endonuclease into bDNA, which is having 5 prime to 3 prime and 3 prime to 5 prime strand, which are joined together, and if we add endonucleases, then there is cutting of DNA in between. See, there is cutting of DNA in between, right? The DNA segment which is within the DNA is removed. Okay, so endonucleases will be cutting the DNA at specific location within the DNA. Okay, regarding the exonucleases, there is cutting of nucleotide at the ends of the DNA. I hope you all are able to understand. Yes, we have regarding the restriction enzymes, we can also call them as uh, nucleases, regarding which we have two major types: exonucleases, which are cutting the DNA at the ends, okay, at the terminals, and regarding endonucleases, these are cutting the DNA in between. Within the DNA strand, there is cutting or removal of the DNA segment, and that is endonucleases. Okay, I hope every one of you are here. Yes.
Yes, sir.
Okay.
Yes, sir.
Yes. So we looked at, see, within this slide, we were trying to understand what is restriction enzyme. Okay. We had first let me yes we had looked at what exactly is restriction enzyme. These are also called as molecular Caesar. As you have seen, there is cutting of DNA, right, and acting like a Caesar, right, and these are though naturally present in bacteria as a defense mechanism against bacteriophage, as a way to protect the bacteria themselves from the attack of bacteriophage, but these can be now used as a tool in genetic engineering for cutting the desirable DNA at recognition site, right? And if we could cut the DNA at recognition site, then we can generate or we can produce DNA which is having sticky ends, DNA with terminals which can join easily. So the collective term used for restriction enzyme is nucleases, right? Collective term or common term which is used for all the restriction enzymes is known as nucleases. We have looked at two types of nucleases: exonucleases, which is cutting the DNA at the ends or at the terminals. Exo means external, outside, right? Terminals or two ends of the DNA are usually the external part, right? Endonucleases are within, these are acting within the DNA. So we have seen, see, removal of the DNA strand in between, right? So endonucleases cut DNA at specific locations within the DNA. I hope you have understood. Now, if we look at the activity of the restriction enzyme, we do have time, right? We still have half an hour, so no need to worry. So, activity of restriction enzymes. We are saying that restriction enzymes will be cutting the DNA. How they are going to cut the DNA? See, restriction and endonucleases will first observe and inspect the entire length of DNA sequence. Suppose, say, like we have this DNA sequence having 5 prime, which is having GAATTC as nitrogenous bases, 5 prime end to 3 prime end, even 3 prime end to 5 prime end, which is the opposite way, right? GAATTC and GAATTC. Okay, so suppose this is the segment of DNA which is to be observed or inspected by restriction enzyme. First, there is observation of the entire sequence, and the restriction endonuclease will find its specific recognition site. Okay, first there is inspection or observation of the entire length of DNA, then later it's could find its recognition sequence, and after finding the recognition sequence, then the restriction endonuclease will cut the DNA. So see, this is the length of the DNA. Okay, suppose eco R1 is the restriction enzyme, it will observe entire length of DNA and find the recognition sequence. After finding the restriction site or recognition sequence, then the eco R1, which is an example of restriction enzyme, will cut the DNA, okay, and forms two cut or two sticky ends of the DNA. See, in between G and A, there is recognition site. In between G and A of the opposite strand, there is recognition site. And cutting of DNA, forming a two easily join. We can easily stick these two DNA, right? As it is cut with the same restriction enzyme. Okay, and whenever we are cutting the DNA, there is cutting of DNA at the sugar phosphate backbone. Okay, so due to production of such sticky ends of DNA, then there is easy joining of the DNA with the help of ligase enzyme. So whenever we are cutting the DNA with any of restriction site, usually the restriction enzymes will be recognizing specific sequence of DNA, and that is usually for eco R1, it is palindromic nucleotide sequence, as we have seen. Okay, so palindromic, the term is important for us to understand. Regarding the palindrome, we can say that in our English words, say like mom, Malayalam, if we read mom from the beginning, from the forward direction, it's mom, right? Even if we read in the reverse direction, even from the ends also, it's mom. If we read the Malayalam from the ends, it's M-A-L, mal, Malayalam, right? Same words can be read in the forward direction and reverse direction. So these words are usually called as palindrome in English, right? Same is the case regarding our restriction enzymes. Restriction enzymes can identify DNA, and they can identify DNA which is having palindromic nucleotide sequence. Nucleotide sequence means nitrogenous bases which are present within the DNA. That is, we have GAATTC. These are nitrogenous bases, right? So these sequence regarding the eco R1, see, it is recognizing in between G and A, right? GAATTC. If we read the opposite strand, GAATTC, same palindromic sequence will be recognized, right? So there is recognition or identification of same nitrogenous sequence, okay, by restriction enzymes. So we can say that restriction endonucleases will be recognizing, will be identifying specific palindromic nucleotide sequence in the DNA. Okay, even if you haven't understood this, we'll have a better understanding. See, we are saying that we have we have restriction enzyme called EcoR1. EcoR1 is one example, one of the commonly used restriction endonuclease or restriction enzyme, which will be cutting at the DNA, which will be cutting both the strand of DNA in between G and A. G for guanine and A for adenine, right? And they are your EcoR1 is having palindromic nucleotide sequence, that is GAATTC. Suppose say like this is one of the vector DNA having palindromic sequence, nucleotide sequence, or palindromic recognition sequence GAATTC, which is having opposite strand GAATTC, which is palindrome, right? The nucleotide sequence or nitrogenous bases are palindrome. If we read in forward direction of one strand, it's same, and if we read the opposite directions, nitrogenous bases, it's same, right? In reverse direction, right? Same is the case. We are having foreign DNA, which is also having GAATTC, and the opposite strand of the DNA GAATTC. So within the DNA, we are having these nitrogenous bases which are forming recognition sequence or nucleotide sequence. So EcoR1, as we said, that it is having its recognition site in between G and A, guanine and adenine, right? Same is the case here, guanine and adenine. Likewise, EcoR1 is having same recognition site in between G and G and A of the other strand in case of foreign DNA. Now, by the action of EcoR1, which is having recognition site, which we usually call it as palindromic recognition sequence, will be cutting in between the G and A, right? And in a way, there is formation of sticky end, two ends of the DNA which can easily join, right? If we are having such kind of product, it's easy to join, right? Woodcutters, the carpenters used to make such kind of L-shaped structures so that they can easily join, right? So same is the case, genetic engineers used to produce sticky ends by using restriction endonucleases and cutting in between, and later on they will join with the help of ligase enzyme, and that forms a new recombinant DNA, newly constructed DNA, which is having foreign DNA and the vector DNA. Okay, I hope you all are able to understand.
Yes,
Yes, so this is
yes,
a recombinant DNA. What we have seen is we are saying that we are trying to understand an example of restriction endonuclease. Okay, restriction endonuclease EcoR1 will be cutting DNA in between guanine and adenine. And we are saying that restriction endonuclease EcoR1 is having recognition site, which is usually the palindromic recognition sequence. And that palindromic nucleotide sequence or recognition sequence is guanine, adenine, adenine, thymine, cytosine, cytosine. Okay, now we have seen vector DNA, which is also having GAATTC, guanine, adenine, adenine, thymine, thymine, cytosine. And same is the case of the opposite direction strand, right? Guanine, adenine, adenine, thymine, thymine, cytosine. This is regarding vector DNA. Now in case of the foreign DNA also, same sequence, same palindromic nucleotide sequence or palindromic recognition sequence is present. See, we have GAATTC and GAATTC, right? And regarding the EcoR1, it is having recognition sequence in between G and A in case of the vector DNA, and that of the foreign DNA in between G and A, right? And after the action of EcoR1 restriction enzyme, then there is formation of sticky ends of the vector DNA and foreign DNA. And this can be easily joined with the help of ligase enzyme, and they forms recombinant DNA or rDNA, which will be used as a tool, right? Which will be inserted or transformed into E. coli. and we can produce different products. So same pattern, this is a diagrammatic representation, and here in your textbook this is represented, right? Action of restriction endonuclease echo R1, where we have the vector DNA and foreign DNA, which are going to cut in between GNA, and even in case of the foreign DNA, opposite direction, right? See, opposite strand also, and formation of two sticky ends. And these two sticky ends can be joined together with the help of ligase enzyme and forms recombinant DNA. This is the diagrammatic representation given in your textbook, which means this part is very important. Biology is having so many diagrams and so many other pictures, graphs. All these are very important. Biology as a science is visual science, right? So that's why uh, we have to look for the diagrams which are given in the textbook. These are very important. Okay, so we can say that echo R1 has palindromic recognition sequence, as it is having recognition sequence in between G and A of the palindromic nucleotide sequence GAATTC. Can we proceed further regarding the naming of the restriction enzyme? Yes?
Yes, sir.
Yes, I hope every one of you are here. Restriction enzymes,
Yes, sir.
yes, restriction enzyme eco R1 is having its own name, right? How it is named. So, regarding the restriction enzyme eco R1, it is extracted or it is obtained from Escherichia coli, E. coli, and that E. coli is having strain of RY13. So, the term eco R1 is name of restriction enzyme, and it is obtained from a bacteria called Escherichia coli, which is commonly present in our intestine, right? So E is the genus name of the genus, that is Escherichia, right? Escherichia is the name of the genus of the bacteria. You are knowing what is genus, right? You have studied about taxonomical category, where we are having species, genus, family, order, class, and phylum and kingdom and all, right? So genus is the E here is the genus, name of the genus, that is Escherichia, and CO is the second two first letters of the second name, that is coli. So CO is from coli, right? CO. So the first two letters of the name of the species is coli. Okay, and then we have R, which is the name of the strain. The strain is RY13, right? That's why the first name of the strain R, and 1 is the Roman numeral 1. So eco R1 was first obtained from E. coli, and it is the first restriction which is discovered from Escherichia coli. That's why we are representing the last letter as 1, Roman numeral 1. So Roman number 1 is representing the order in which the enzyme was isolated from the strain of the bacteria, that is E. coli. So from E. coli RY13, we have isolated eco R1 as the first restriction enzyme. So this is how we are naming the restriction enzymes. Okay, I hope every one of you are able to understand what we had discussed so far. Yes.
Yes, sir.
So this is important. So without taking much time, we have still 13 minutes left. We'll just look at how we can separate and isolate DNA fragments. See, we are playing with the DNA, right? In genetic engineering, we are playing with the DNA. We are just constructing new DNA, and we are deleting some parts of DNA. And in a way, we are doing so many experiments regarding DNA. So how we are going to separate DNA from the cell of the living organism, right? And how we are going to isolate the DNA fragments. So you have already studied about extraction of DNA, right, from different plant sample as an experiment in your first term. So usually after the extraction of DNA, then in order to purify the DNA, we use a technique called as Agarose Gel Electrophoresis. And whenever you are studying about any term in biology or any other science, we should look at the terms, and we should try to split the term. Regarding agarose, the term is ending with suffix ose, which means it is a carbohydrate. So agarose is a carbohydrate, right, which is obtained from algae, right? We'll discuss in detail later in the slide. And it is forming a gel, right? Agarose is like corn flour. If we cook it and then if we cool down it, then it will form a gel. It will solidify. And in the process of separation or purification of the DNA fragments, we are also using electricity. And that electricity will be helping in movement of the DNA. And in a way, according to the size of the DNA, mass of the DNA, charge of the DNA, we are going to get our desirable DNA. Okay, so separation of DNA or purification of DNA, the desired DNA can be obtained with a technique or with an experiment or with a protocol called as Agarose Gel Electrophoresis. Okay, you should keep this term in your mind. Okay, how that is done? It is done with the help of this setup. This is an equipment which is used. This is typical equipment which is used in order to perform agarose gel electrophoresis. And here what you can see here is the source of electricity or voltage source or power supply. So power supply is here. This is the stabilizer which will be receiving electricity from the main source, and it is having two electrodes, right? We all are knowing that from the power supply we get two electrodes, right? Two charge, negative and positive, right? Here in case of our agarose gel electrophoresis equipment, we are having cathode terminal, which is the negative terminal. Okay, and we have anode terminal, which is the positive terminal. Okay, while switching and that cathode and anode terminals are attached with a tank, right? This is a tank, a small container, right, where it is having a platform in between. So this is the gel tank in which we are going to keep the gel. Okay, so gel is first produced or first prepared separately by boiling, and first we'll be taking water, okay, distilled water or double distilled water. Very clean distilled water is taken, and in it we are going to add agarose. Agarose is a carbohydrate which is available in the lab in the form of powder. And after adding the agarose into the distillated water, and after boiling and mixing, and then in a way we can prepare agarose gel by cooling it on this casting tray. So this is known as casting tray, and casting tray is usually uh, for preparation of the gel. And after preparing the gel, then we can while preparing the gel we are also going to make wells. See, we are having wells here means they are tiny depression which are prepared with the help of an instrument called comb. Okay, the comb is having tooth, okay, similar to the comb that we use for untangling our hairs. We also have comb which is used in the genetic engineering lab regarding the gel electrophoresis, and that comb will be making tiny depression or tiny pores called wells, and into which we are going to load the DNA. Okay, once after all these setup is complete, then after say like switching on the power supply, then we are going to run the DNA. After loading the DNA into the wells, then on the basis of charge, on the basis of charge, mass of the DNA or length of the DNA, then as the electricity passes through negative terminal to positive terminal, then usually the valves are located near the negative terminal or cathode terminal. Okay, and from the negative terminal to positive terminal there is migration of the DNAs according to size, according to charge. Okay, and in that way we are achieving the gel agarose gel electrophoresis. Agarose, why? We are using a powder which is mixed in the water and to prepare the gel, agarose gel, right? And into the agarose gel in the wells we have added DNA, right? And as the DNA are migrating according to the charge size, we are calling it as electrophoresis. There is movement, migration with the help of electricity from the negative terminal, that is cathode, to the anode, positive terminal. In that way, suppose this is our desirable gene, that DNA fragment is desirable gene or DNA, then we can cut this DNA and we can amplify, we can make this DNA into thousands of number with the help of PCR. I hope, though this is new to you all, I hope you are able to understand general idea about what our typical Agarose gel electrophoresis equipment looks like. Can you all understand? Yes?
Yes, sir.
Yes. Now we'll stop by looking at this important part of your genetic engineering. See, we said that there is formation of a gel in which we are going to get DNA fragments, right? And in your examination, this question may be asked. Okay, so this diagram is from your textbook and it is showing the gel which is already run, right? Here we are having the wells into which we are going to add the DNA with the help of dye ethidium bromide, which will be showing us the bright orange color. Okay, and see, largest band, DNA bands are of different types. DNA fragments which are heavier are not moving fast, right? See, lighter DNA, which are small DNA, they are moving fast and they are near to the positive terminal, right? And we are saying that there is running of the DNA, or the wells are located near the cathode, that is negative terminal, and DNA is usually negatively charged. So that's why we are adding the DNA into the cathode or negative terminal, and there is movement of DNA from negative terminal to the positive terminal, that is anode. Okay, so with reference to this we may get question. See, DNA bands, or these are DNA fragments, okay, cut portion of DNA. These DNA are cut with the help of restriction enzyme. We have discussed a lot about eco R1. Suppose if we cut the DNA with the eco R1, then eco R1 will be giving different types of fragments of DNA. Okay, and those DNA, after adding into these well and after running with the help of electricity into this gel, then there is separation of the fragments according to the size, right, according to the mass. So smaller is the size and smaller the weight, these are migrating or moving from the negative terminal to positive terminal faster. Regarding the largest DNA and heavier DNA, they are not going to migrate, they are not going to move faster, they are going to move very slowly. And we can say that now DNA is negatively charged and it is moving from negative terminal to positive terminal. Okay, are you all able to understand what we have studied so far? Yes.
Yes, sir.
And in your lower classes you have studied that, say like in class
Yes, sir.
11th, you have studied about gelidium and gracilaria. Gelidium and gracilaria, these are usually algae, and from these two algae we are going to get agarose gel, agarose carbohydrate, and that will be used for preparation of agarose gel. Okay, so agarose is a carbohydrate which is used for preparing the matrix or the gel, in which is used for gel agarose gel electrophoresis, is obtained from gelidium and gracilaria. These are algae. Okay, and we said that there is cutting of the DNA with the help of restriction enzyme, right? And agarose gel electrophoresis is helping in separation of the DNA fragments on the basis of size, right? Larger fragments are moving slowly, right? Smaller are moving faster. And molecular weight, DNA is usually weight, the size of the DNA or the DNA's fragments are categorized on the basis of molecular weight. Okay, and even on the basis of charge, DNA is negatively charged, so there is movement of DNA from negative terminal to positive terminal. Okay, so negatively charged DNA fragments will be separated or will be resolved or purified by forcing them to move from cathode to anode, that is negative terminal to positive terminal, with the help of electricity through the gel matrix. Okay, so this is what you need to understand so far. Okay, and in the tomorrow's class we'll try to understand visualization of isolated DNA fragments. Okay, I hope every one of you had understood what I've explained so far.
Yes, sir.
Yes, sir.
Thank you for attending this class. I'm a bit
Yes,
worried
sir.
about your syllabus. That's why I've taken this extra class. Okay, I hope you all are not bored to listen to what I've taught so far. And I'm expecting that these information are quite useful for you all. Even if you haven't understood, who knows, if my live recording had went properly, you can watch that video again through YouTube. Okay, and even you can refer the notes which I had already uploaded, and you all are having access to those notes, right? So you can just read those notes, you can refer your textbook. I hope at least I'm here to help you all to understand the concept, though the concept of biotechnology is totally new to you all. Okay, I hope I'm making some difference, some changes for your supporting you all with little contribution. Okay, thank you.
Thank you, sir.
Thank you. If you are having any doubt, you can WhatsApp me, okay, in my number so that I can
Thank you,
help you
sir.
all. Thank you all. Thank you all. Have a good day. Have a good. Now it's not day also, right? You have attended. It's almost 5 o'clock. Okay. Thank you all.
Thank you, sir.
Thank you.